In our vitamin C analysis experiment, we're going to be using a redox reaction that is based on organic compounds and not inorganic compounds where you had the charges changing. These are covalent compounds and there are no full +1, +2 type charges. Anyway, for carbon-based compounds, reduction means gaining bonds to hydrogen or losing bonds to oxygen. And then the opposite for being oxidized. Something is oxidized if it's gaining bonds to oxygen or losing bonds to hydrogen. So let's look at this example. In this example, we see that this is methane gas, like natural gas, and it has bonds to hydrogen here, but it's getting bonds, it's losing those and going to oxygen here. So this compound is being oxidized. And what is oxidizing it? The oxidizing agent. And then we can also say the opposite, that the oxygen is gaining bonds to hydrogen, so it is being reduced. And that means that the other reactant is the reducing agent. So that's an example of a redox reaction. We'll see ours here in a second. So we talked about how these are kind of opposites, with the oxidizing agent is being reduced. What about the word antioxidant? You've heard of that. Vitamin C is an antioxidant. An anti means opposite. Oxidant means oxidizing agent is an oxidant. That makes sense. So an antioxidant is a reducing agent. And the reason that's important is that in your body, sometimes there are oxidizing compounds or species that will react with your proteins and do damage. And if you have antioxidants present, those oxidizing molecules or ions will react with the antioxidant instead of damaging a protein. We will be doing a technique called a titration. All titrations involve a chemical reaction, and our reaction just happens to be a redox reaction. All titrations also involve an analyte, which is what you're analyzing. And the analyte, we don't know how much there is. It has an unknown concentration. Our analyte is our vitamin C solutions. To have a titrant, which is the other reactant. Our titrant is DCIP. The idea is that we'll start with a sample of our unknown analyte, or vitamin C, and then we'll add the DCIP drop by drop. This reaction will start to occur, and we'll keep adding the DCIP until there's no vitamin C left. And at that point, based on how much of our titrant that it took, we can figure out how much of our vitamin C was there. Well, how do we know when the vitamin C has all reacted? How do we know when this reaction has stopped because all the vitamin C is gone? Well, we use something called an indicator. An indicator is something that causes a color change when all of the analyte is gone. So we'll be adding the DCIP dropwise until we get a color change, and at that point we can look at how much DCIP it took and figure out how much of our vitamin C was there. When you're adding the titrant dropwise and you get the color change, that point is called the end point. When you see the color change, the titration is over, and you will look at how much of the DCIP that it required to get there.